Okay, guys, good morning, guys, of grade 9. Today, inshallah, we're gonna pass to chapter 6, section 1. So, reading chapter 5, we studied the heredity and we studied the Mendel experiments and how he could or how he was able to explain the results he got from his first and second experiment. And we know he opened the gate of the genetic science, or he the one who established the main principles of genetics. But we now are gonna go beyond Mendel, that we are gonna pass inside the cell. We'll find what are the genetic material, what is the structure of the genetic material which are responsible for these different phenotypes or these different alternatives of shapes that we have in different living areas. So here we're gonna talk about what does DNA look like, what is the DNA. All of us know now that genetic material inside any cell is known as DNA. So what is DNA? Actually, DNA stands for deoxyribonucleic acid. So deoxyribonucleic acid. So nucleic acid here, as we remember last year, nucleic acid is one of the macromolecules that already build any cell. So whatever the kind of the cell, starting from bacteria to the human, okay, we have DNA in our cells. So, and the DNA in all living organisms somehow it have the same basic structure. So why? Because actually all the DNA they apply the same function, which is storing and passing the genetic material or the genetic information. So if we look closer here, what the DNA is made up of, so we're gonna find the DNA is made of smaller units known as nucleotide, or it's made of nucleotide. So if we keep you look at this diagram here, guys, you could observe easily that that's how the nucleotide look like. Okay, so you can find that the nucleotide here, that is all this is nucleotide. Okay, so the nucleotide is made of three subunits or three small units. Okay, made of 5 carbon sugar. That's why I arrange it in a pentagon shape, because it's made of 5 carbons. So 5 carbon sugar, or the ribose sugar, and phosphate group. And here in that side there is a nitrogen base. Nitrogen base, it means a certain chemical structure that includes nitrogen in its structure. So this nitrogen base, okay, could be one of four. It could be alternative. Okay, what are these four alternatives? A, which means abbreviation for adenine. T stands for thymine. C stands for cytosine, and G stands for guanine. So here we have this four, okay, nucleotide. That means we have four different nucleotides. Yes, we have four different nucleotides. They just different in the nitrogen base, but the sugar and the phosphate, they are exactly the same. So here are these four different nucleotides. They could be found in DNA in different ratios and in different arrangements. That's which create the huge diversity in the DNA, that there is no two DNA are the same. Even the DNA of identical twins, they still have some differences. Okay, so the differences are gonna back to that different arrangement of these nucleotides. What is the code will be. So here, this the code depend on the arrangement of these different nucleotides and the amount in which these nucleotides are represented in a certain sample of the DNA. So here, like this, guys, okay, we know what is DNA and what DNA is made up of. Actually, how these nucleotides are arranged, or how they look like, how the chromosome, as we know, it includes the DNA, but how the DNA itself look like. So we know, or nowadays, everyone, if you ask him how the DNA look like, so generally or simply, you just draw the most famous, okay, drawing that here it's look like this. Actually, this shape is known as the double helix. Okay, so we know that's the shape of the DNA, but actually how scientists be able to reach this shape, or how DNA is arranged in that matter to look like this helix. Okay, so that's what we need to answer, okay, in our section here. So here, let's back to the history and find how science start to gather info about the shape of the DNA, or how the DNA look like. So the first scientists ever here start to do an effort in that field, Chargaff. So Chargaff reached a rule. There is a scientist, his name Erwin Chargaff. He started to use to make a kind of chemical analysis for the different samples of DNA of different living organisms, and he always get the same result each time. That always the amount of adenine, that means the base A, equal the amount of thymine, the base T, and the amount of guanine, the base G, equal to the amount of cytosine, which is base C. So A equals T, C equals G. Okay, that always the result. So he said it's a Chargaff's rule. Okay, And like this, that the first thing we know about the structure of the DNA, that always adenine pays equal thymine and cytosine equal guanine. That was not enough to have a very clear image about how is the structure of the DNA. So scientists keep researching and researching and making a lot of experiments to find what is the structure of the DNA. So then we get a famous scientist. Her name, sorry, is Rosalind Franklin. Actually, Rosalind Franklin considered the first one who able to describe the DNA as double helix. And actually, there is an exciting story beyond or behind Franklin. Actually, Franklin was a famous chemistry scientist, okay, that start to work in a hot field of science like searching about the structure of DNA. In that time, society was not that helpful for females or women to work in such fields. Okay, so it was not that familiar with her colleagues, and actually her post in the lab was not that helpful. Actually, he started always to put her away and something like that. So actually Franklin used a technique called X-ray diffraction. That means it's just apply high energy X-ray on crystals that contain DNA samples, and then they get the images of the DNA and start to study this and analysis how these images are formed, and try to find the angles, the position of atoms, and like this. And actually she did that for a long time and tried a lot of experiments until actually in 1952 she got a brilliant image of X-ray diffraction of a certain piece of DNA, which gave her a very strong hints about the structure of the DNA. In that time her post stole, okay, where actually he took these results without her known and showed to other two scientists who work in the same field also. In that time they searching for the structure of the DNA also with Watson and Crick. Watson and Crick actually they don't know that is the work of Rosalind Franklin. They saw that her post, okay, uh, that his work, so they share with him to keep working on it, and they already save a lot of time instead of all this work which done by Rosalind Franklin. They start from the next step, which make them faster to publish their research about the double helix shape of the DNA, and they got Nobel Prize. But actually, later they said that the one who deserved Nobel Prize was Rosalind Franklin, but unfortunately, Rosalind Franklin did before, okay, anyone know that she did that. We know that later, okay, after her death. So that means Rosalind Franklin already used the X-ray diffraction, and she was able to know that the DNA is made of two strands, and these two strands are twisted around each other, they forming the shape of helix. And then Watson and Crick, they used the data of Rosalind Franklin, and they keep researching about the DNA until they were able to build a model that represent the position of each single atom or the position of each part of the DNA. And they call this model the double helix model, and that represent, okay, how the DNA look like and how that could fit with this function, or how that fit that the DNA could be replicated or could be passed from one cell to another. And that's why they get the Nobel Prize about this. So next time, guys, inshallah, we're gonna know what is the model or how the double helix model of Watson and Crick look like, and what is the exact structure of the DNA. So please study this part well from your textbook, and if you have any question, you can contact me anytime. Thank you and goodbye.